we go. So once again, welcome. Let's talk about all that we achieved for the research infrastructures this year. Of course, it's no longer just ARISE, so you'll hear from the teams about ARISE, but also eDentity, some BGE, BMD, and at the end, even our latest program, ABM. So without too much ado, let's share some knowledge. Like I said, we have multiple programs now that nicely click into each other. Most of them are around species recognition and how can we build up our capabilities around identifying species in the field or basically anywhere rapidly with the latest technology, but also how then can we transfer to monitoring and actually getting data insights about our biodiversity and ecosystems. The teams are quite expanded, as you know, so we have a whole sample to DNA ID section, we have a sensor to AI ID section, and you'll hear all about all of these. Also that we can use and basically have end-to-end workflows for research, but also hopefully for the wider community. That means first and foremost, we need to build that reference database. Without the knowledge, and that's why we do it, we can't do the identification. So we really need to have the basic knowledge, the reference databases, a specimen, an image, a DNA barcode, sounds, traits, observations, and of course the taxonomy all together and harmonized into a large data system. But let's start in the field, and I'm going to hand over to Emil from the UvA to tell us more about that.
Thanks, Elaine. I'd like to give an update about what we did and are planning to do in the monitoring and demonstration sites. So part of it is a bit of repetition of what people who've been here before recognize. Perhaps you can go on to the new slide. I think I can't control it. Yeah. So the purpose of these sites is to show how different sensors work, also how to test and figure out how these things work and what they actually observe, and how beyond identifying species, these sensors can also answer other questions. And I would like to emphasize also the third bullet point, which is the idea is to connect to colleagues, that is other scientists in the field, but also stakeholders who want to know something about these sensors. So if anybody is listening to this presentation, you can always reach out to ask something about the equipment that we have in the field, or some ideas about perhaps new sensors that may be deployed. Having said that, let's move on to see where we do our monitoring. So we have done deployments of all these sensors over the past years, and some of these are still in the field or are going to be placed in the field again in the coming spring. Apart from the variation of sensors that for audio imaging, a radar, and some environmental variables like temperature and soil temperature, there is this these numbers at the far right that indicate that there are in fact many devices and many data, much of which is stored but not processed yet. So we have a kind of backlog in identifying the species that is being the species that are being in those data and have to be discovered still. So that's also part of the job that we are at. Can we move on? So we collected the data at many sites, and the red dots are very experimental sites, also people's gardens, and the black dots are the monitoring sites where there's more equipment and there's more long-term monitoring. Let's move on to the next one. So it was the past, so these are the core monitoring sites, I would say. And for the coming year, we plan to extend the number of sites to one area north of Amsterdam in the PT region of Waterland, where we want to do some acoustic monitoring related to a PhD project. A site in the Dune area managed by the water company Dunea, where we will place a radar and do some related acoustic monitoring and test insect monitoring. I will see a slide later on that. and a site where we would like to do acoustic monitoring related to, let's say, public outreach in community gardens in the region of Amsterdam. And there will be one special site which is different from all the rest, because that's the first really aquatic monitoring that we will look into. That is with by using a sonar, looking at underwater vegetation. And this will first take place in Markermeer. So the purple dots are what we're going to do the coming year, and I will highlight a few ideas or plans that we have. This is quite concrete. We will, in Dunea, as part of our work there, test again the automated equipment to look at moths and compare these with the more established equipment, which is like this LED buckets. So do some manual identification and compare these with machine-based identification, and also compare that with diopsis and do something similar. Yeah, thanks. Let's move on to the radar. And by the way, these projects are usually centered around student research, because that's for us the easiest way to have enough hands to process the data. Something that's going to be a bit long term is this radar. I've here put a photo of the radar looking at birds and insects that is positioned at Attis, but a similar machine will be placed in the Dunea terrain, and it has a camera that also photographs birds that are passing by. So the identification of the actual birds flying around is possible by image, but also by linking to the captures at the ringing station and the recordings by acoustic devices. So this will be combined there. In the Lake Markermeer, the sonar is in fact measuring, similar to the radar, a signal that is not identifying at the species level per se, but we'll try an image like the blue one at the upper right is recorded usually, and this is translated into vegetation types or specific species by doing experimental work, like shown at the bottom, where you can see the actual photographed or manually recorded species and relating this to the shapes that are seen in these red and green pictures. So this is a project that will start coming year, but it will take several years before I think we'll have some useful operational machinery there to identify species with sonar. Then I would like to highlight again that our biggest monitoring network is with camera traps in the Amsterdam dune area, operated by Waternet, and this is still continuing. We have a focus on larger mammals like the deer population and the rabbits and the hares, along with predators like fox. Let's move on. So this is an impression of the type of images that are collected and the type of species that we see. But let's move on to another slide. We will expand and still try to do different things and with this equipment. So we looked last year into the vegetation classification using the same network and see how far we can get. We will continue to look into this and also do some phenological observations based on these cameras. Let's move on. Also, we have tried different setups of the cameras, like this one, which we call a selfie trap for arboreal species, like these woodland birds, but also especially the squirrel was a target species. It started to visit these camera traps recently, and we will compare them with other setups and acoustic monitoring to see what the different devices monitor and how they complement each other. Let's move on to another. We'll experiment over the coming year, especially with different setups in the dune area, like this example where a camera is downward looking. And finally, another example of the added value of combining acoustics and imagery at the same location. So if, again, this was my last slide. I would like to emphasize that if you have an idea about a different sensor that was not listed here and for trying it out somewhere, we are very open to get in touch, as well as questions regarding the sensors and experiences that I've presented here. Please get in touch.
Thanks, Emil. Good morning, everyone. I will now talk a little bit about the DSI team, the species identification part of ARISE. So let's dive in. An actual update on some numbers. It's always nice to show. So since ARISE launched, we had a lot of applications to use the system. So this number of 126 users is probably already a bit outdated. So there's a lot of interest in using the system, which is really good. We have more than 1,500 deployments in the system, which means a device recorded at a location during a period. Over 40 million images. To be honest, most of them are currently Insta-camera images, since that's what we started off with. But also more and more wildlife camera images are there. We now have 8 algorithms to process images and 2 for audio, and that number is growing. We have analyzed over 40 million media items, and in those media items we found more than 100 million potential insects, birds, or mammals. And this will ramp up very quickly in the next year. So a lot is going on. So let's some highlights. We did a lot of UI updates and are continuously improving the user experience because we're starting with a prototype, which is not always great for user experience, but it's getting better and better. It's now even easier to add algorithms, so it will become quicker to add more algorithms. We've recently added 4 for wildlife cameras and 2 algorithms for collection data. I'll get back on that on the next slide. A lot happened for the system robustness and stability, because as more people are using it, we are finding more bugs in the system, pun intended. One nice example of that is that one user was able to start thousands of analysis jobs overloading our system. So now you can't anymore. That's fixed. We limit the amount of jobs so you can start per user. Next. At the same time, we also invested some time in creating load tests for our system. So we find bugs before you do as a user. Then we've added project management. We'll get back to that. And also we have an elaborate wireframe design, which is basically a duplicate of our system, which we can use to design new elements and test them with users before we actually build them. And that's really great to have. It will really speed up the development in the future. So this is a current screenshot of the dashboard as it currently looks. One thing you notice is that we can now show system messages on the top. So last week, Snellius, our computing platform, was out for maintenance. So those kind of things we can now show on the system. And also the menu structure has changed. Oh, if you can go one back, Elaine. Yeah. So on the left is now quite a clean menu. And if you click on Media Browser, which is the next slide, it opens up a sub-menu, which now shows a button to upload media, to browse, or start an analysis. And a lot of other things have moved to manage, which is on the next slide, which is where you can now manage your sensors or locations, deployments, datasets, and algorithms. It's more intuitive now. If you don't know the system, it may still be overwhelming, but there are tutorials. Also, we are also working on documentation as we build this. So a little bit details about the algorithms we recently added and will add. So Tetris, Vertigo, and Tetris and Tenerife are developed within Naturalis and can be used to process images from collection material. I have an example of that in the next slide. We have added the SpeciesNet, which is a big algorithm developed by Google employees, which we can use to process wildlife camera images. And very soon, we will also make the Naturalis multi-source model available in ARISE. This is the model that is behind Obsidentify the app that you can use. And we will link to it from this Arise system. So here a quick example of the type of images that can be processed by the Tetris algorithms. So, and this is an example of one photo from the Amsterdamse waterleidingduinen. And if you look on the right, you can see that we actually process this image with 5 different algorithms. And this will, so if you have data, you can now use, so one of them is Tetris Tenerife. That's not really meant for this type of data. So someone run an analysis job there, but the other 4 can now be used for wildlife camera images. Yeah, we also spent, in the last half year, we spent a lot of time on the sensor portal, which is the part of the application that allows you to connect sensors and manage them in the field, and also the data that comes out of them. So you can manage, set up projects, add sensor models, different devices or sensors, and also register the deployments in the field. Then for each sensor, you can upload media items. So we've done a lot to improve the user interface for that on the front end, but we're also working on the back end so that you can use scripts. So if you are a power user and you don't want to use a front end, we are working on a software development kit, so you can locally use this to push data to Arise. And at the same time, we are working on support for 4G enabled sensors, which you can configure in the portal, and then they will upload automatically via 4G, automatically even more. So if we dive into that a little bit, this is a very small demo of the upload process. It's now more intuitive. So the first, it's a wizard. So first you select a device, in this case a demo camera. Then on the next step, you select the deployment of the device. If you didn't have one yet, you can click create deployment, and then you will see this in the wizard. So you can add a location and a period. And then last but not least, select the media items. So this is an example of the process to upload data for a device, if the device has an SD card, for example. So you can now manage your projects. So you can manage members, you can give roles to those members of a project, and each role has different permissions. An example of that is on the next slide. So yeah, it's very straightforward, but it's quite crucial in setting up projects to collaborate. So here you can add members and give each member a role. Yep. Then in the next 3 months, we will focus on connecting to a new compute cluster. So currently we are relying on AWS, Amazon, or the Snellius, mostly the Snellius compute cluster at SURF. And we designed it in a way that we can also connect to other compute clusters, which we now want to demonstrate by connecting to a compute cluster at Naturalis. And in the future, this feature will also allow you to connect maybe to your own compute cluster in your institute, if you have one. We also want to add 3 more algorithms and work on user feedback from you, the user, and improve our system based on that. And last but not least, also support 3 more sensor types. So currently we do not fully support the song meters yet. So that's something we want to work on. The 4G wildlife cameras, I think we are 70% done by connecting them. And also the Netbox cameras should be supported in 3 months from now. That's it.
Thanks a lot, Jacob.
Yep.
Awesome. We need to speed up a little bit because there's a lot more teams waiting to say something. So let's go.
So for the sampling team, I will tell about the numbers. Of course, we like the numbers. This is a cumulative number of specimens collected in Arise, which is in the reference database. I think about this year we have added about 6,000 species identified specimens to that. Of course, everything is registered, sampled, the metadata are registered, but this is something we always have done. We added, if you go one back, we added now also that we do collection mining. So before it was mostly fresh specimen. This year we have collected about 1,000 specimens from the museum collection, and we will keep doing this next year. Everything is subsampled. and imaged, of course, and this is all stored. So these are the current numbers when you go to the target list of the different taxa. This is based all on the Dutch species register, everything which is registered there, but it also includes many species, for instance, that are exotic or not really native or established. If you go one further, here you see actually the percentages if we would zoom into the established species and also include some data sources that we're not yet able to really connect. But if we do that, you see we are really close to reaching the targets, particularly for the plants. We know we include all the hybrids. With Animalia, we're getting really far. And the fungi, of course, they are problematic, but many of the fungi are not really able to collect, but we did not exclude them now from the list, but we should probably next year. These are some further numbers. I would like to highlight that there's from this large number of 146,000, about 22,000, 23,000 that are really identified to species level. All the others are from bulk samples. So 123,000. And of course, we need the experts in the field to really identify all the stuff that we have sequenced and maybe have an ID. Still, we have about 13,000 first priority species, which are the species that have no barcode at all worldwide. And you see a list of the larger groups in there that we're actually seeking more expertise. As I said, we do collection mining, still identifying for bulk specimen and validate the generated barcodes. That's something we would really start up next year. And if there are any questions, please reach out to us in the info email, of course. So we help out also with the data problems that we face. Of course, we're not only collecting for ARISE. So I mentioned the 123 hand-sorted ARISE specimen, actually from bulk specimen. This is a lot. There has been a lot of work. These were collected from traps. Something we are working on the next few years is a biosorter pipeline. I will not go into detail about that right here, but I would like to emphasize to mention it to you. So this is actually coupling devices that really are able to sort bulk specimen automatically based on digital species identification. They will do imaging, and we will do high throughput sequencing on it, so that we actually have an automated system, a biosorter pipeline, to select specimen of interest, as I call them, that we can actually reach out to the taxonomist. Okay, so this is coming up in the coming years. Besides that, in eDentity, one of the main aims is to do a collection of eDNA, environmental DNA from the air, water, and soil samples. I will also not go into close details on that, but the main aim is to collect 70,000 specimens. The numbers are given here for the 3 different groups. From all over the Netherlands, to create DNA fingerprints, so we can actually see which DNA barcodes do we know from the ARISE database and which barcodes do we really not know what it actually is. But it is a qualitative measure, so we cannot say about the number of the species that is in there, so the number of one species, but we can say the number of different species. The focus here that we do have at this moment for this month actually is that we would like to have a null measure for the urban public area. We will sample about 4,500 samples from all over the Netherlands. Here you see blue and red dots, which is a measure for the number of samples that we will collect from these places. It's 184 locations that we will visit parks, meadows, and single trees, and we will collect samples there. Last week, in 1 week, we collected already about 300 samples, and we will keep doing that. And hopefully this month, we will already get really far in mapping the entire Netherlands, at least in this scale. And of course, we keep doing that next month.
Yeah, okay. So then for the DNA apps team, we develop applications for data management and validation for the DNA workflow. And we have been mainly working on the ARISE platform, the target list, and the DNA dashboard. So to start with the platform, which is now live. And this combines or gathers all tooling that we all have been working on the last 5 years in one place. So for example, the sample to DNA workflow, you find the target list, the ADA sampling tool, and the DNA dashboards. And for the sensors to AI workflow, we have a list of all the AI models that we currently support, and a link to the platform that Jacob just showed. And anyone can now push the button Get Started and request an account. And I believe a lot of people already did. There's also quite an elaborate help page in the menu there. So if you have any questions, please visit that first. And then for the target list, we did some technical things. Yeah, maybe go to the next slide. Because we have, of course, the species status that tells collectors whether we want specimens for a species. But that's not only displayed here, but also in the ADA sampling tool. And they are now using the same source for that, so they are always in sync. And we added, you can click once, added a priority label, and that's for species that don't have any barcode collected yet. And no barcode and no specimen collected, sorry.
Worldwide.
Yeah, so nothing yet. And for the collected column, previously we could only display specimens registered in ADA here, but now we can also add the ones that are registered to a metadata sheet, and also some legacy data. So it's more so already to the DNA dashboard. Here you can find all your metadata and the lab results if they are already in. And what we added is the match. The match species matches. That is if we have a sequence, we look into the global reference database, BOLT, and see if it matches with the identification that the collector gave us. So if that's the case, on species level, we give it a species match. Otherwise, we look to family, if the family is correct. And if we could not find that match, then we say it's a no match. Okay, next slide. So of course, we also like to know how much the tools that we develop are being used. So that's why in mid-September, we added the analytics tool. Of course, it's just data, so you never know what's behind that. But I was quite surprised that people spending on average 8.5 minutes on the platform. So tells me that they are really using it. So that's quite nice. Just good to know. So I think I already told this, so maybe we can move on. So next quite important step for us is next year we are going to work on updating identifications. We are of course working on a validated species reference database, and if the collector for some reason wants to update the identification, we want to know. So you're looking at the design now for this. So you can choose another species and give a motivation. And if someone choose DNA analysis as a motivation, we also display a warning, because sometimes you really have to know what you're doing with DNA analysis. So that's was an important element for us in that. That's it.
Thank you.
Kevin?
Well, while we're on the topic of DNA, so first, of course, some numbers. Machines are running in the lab, and we've been processing samples at a steady pace. So we now have passed, like expected, the 100,000 samples in the lab. So that means more than 100,000 DNA extracts were generated from specimens provided to us by the sampling team. PCR rates are almost 90%, but if you do some quick maths on 100,000 specimens, that still means about 10,000 specimens that have not generated anything yet. So that will be the focus for next year, to redo those samples. But we also see more and more projects coming in outside of ARISE. So a lot of researchers, especially inside Naturalis, are now aware that we have these pipelines and they want to use those. So there have been now about almost 30 projects with more than 11,000 samples that have been processed in the lab at Naturalis using the workflow we set up. But more and more, we are working now towards incorporating those into the workflow we do in Arise. So these plates will come into the batches we already process, so they're not processed separately anymore. But it also means that all the data will come together in BioCloud, etc. We've added some extraction protocols, so we now have a working soil extraction protocol and a working water extraction protocol. Again, a fraction of the costs of commercial kits, which is really nice because this is what we need to build further on eDentity. And in the next slide, you'll see that we're getting there. So this is the DNA meta barcoding workflow for the different sample types, so soil, water, and air. And as you can see, for soil, we already have the whole pipeline up and running. For water sample, we're still working on some sampling protocols and some tooling around that. For air samples, we're also still working a bit on the extraction protocols, but basically the amplification and the sequencing is the same for all three methods. We might use some different markers, but it's still a bit of an open discussion at the moment. And so as an example of what's happening in the air eDNA domain, this is some work being done by the Hogeschool Leiden. They are using all these different sampling tools, basically machines, to vacuum the air around them, collect tapes or filters with DNA, and all these machines probably slightly different. So there's some large-scale comparison going on. One of the areas where they do this is the Hoenderdaal Zoo, if I'm correct, which is a nice location because we know what species are there, and they're pretty easy to distinguish from the real Dutch biodiversity because they're exotic animals. And so you have a reliable source of air eDNA that you can actually test. And looking also at like sample duration, preservation, etc. So this is ongoing. On the other hand, what we have also been doing for, and this is especially important for Arise, we had all this legacy data because we've been sequencing for 15 years. So we've now finally sort of completed it, and there's almost 30,000 records now on Bolt, representing more than 9,000 species, Dutch species. And 22,000 of these are complemented with at least one sequence. So some have multiple barcodes. So also there are some specimens we should probably redo. We're sort of pushing all this data now and making it public so that all of this is available to the public, but also counted toward target list, etc. And on the topic of publications, Westerdijk will never disappoint us with their wonderful publications. So they have now published this new paper. There's a link in the screen where they did basically a reevaluation of some species complexes in Fusarium, which are always accompanied by these wonderful plates. If you have a rainy afternoon, there's this nice paper waiting for you. And I think that's it. Well, yeah, of course, the outlook for next year, full on production. So we're scaling down the barcoding a little because we're trying to focus on redoing the specimens that didn't work in the first time. And that requires some hit picking. So there's a little more work than just taking in plates from the sampling team. And then for metabarcoding, we hope to do 7,000 soil samples and get the water and air up and running. And then we'll also start with the genome skimming. So about 1,000 specimens are planned, basically picking up where BG left off.
Yeah, because BG is finished.
Yeah.
Technically speaking. Right.
Thank you. So bioinformatics, let's start by the DNA processing. DNA processing comes after sequencing, where we process the raw data and provide a single barcode for each specimen, or sometimes more barcodes. We have been focusing on robustness because there have been an issue with the input data. So we have added additional quality checks for the input data, also extra validation step. And we have improved our reports, and we have highlight potential discrepancies in the report as well. And the extra validation step occurs just before sending the data to BioCloud. So now it's not automatic, but there will be a manual review, which actually takes a lot time that having to delete the data, incorrect data in the storage or in the database or even in BioCloud. So it will be more efficient. Of course, we made new barcodes, so we'll lag a little bit behind the sequencing because some of them are still in validation, and we have more sequence, 10% from last 2 months ago. We have a bit more than BioCloud, of course, because some of them are not yet. They are still waiting to be validated and not yet released for BioCloud. The taxonomic identification, so that it's one of the validation steps that occurs after we send the data to BioCloud. So in the summer, we work on a new system using Galaxy, which is a platform that researchers can run analysis, and we use it programmatically as well. So it works, but we have been competing for resources with researchers. But this is going to change because we're going to have more servers, and as well there has been some optimization regarding our tool. And this identification sequence identification pipeline is using reference libraries, so from GenBank or BOLD, and so you've seen that for the metabarcoding there will be more markers from now. Currently we have been using mostly CO1, but we want to be ready for the additional markers that coming from metabarcoding, and also of course we need to do also some curation and some additional to normalize the taxonomy in this different from these different sources because they provide different ways, different taxonomy. The target list. Well, no need to present it, but you probably don't know that the bioinformatics teams is responsible in providing the species list and also some of the numbers in that table. And we have a pipeline for that. The pipeline will run monthly, and it will still do that for the species list and the public barcode. But now, since we have the collected column as well, we refactor this pipeline to run daily. So you won't have to wait weeks to see your specimen in the target list. Now it should take like 2-3 days because there is a chain of pipelines that need 2 triggers, but yeah, it will be a lot more faster. And also we had a manual step of updating this target list even after producing the table. But now thanks to Victor, our data architect, the pipeline can push directly to the database and later to the interface, to the online interface. So the pipeline is offline, but it's almost there's still additional checks to add because it's going to be all automatic. So I want to add more validation. But I have updated the data yesterday, so I run it manually, and so now you should see the most recent validated barcodes and your latest collected specimen on the interface. And we have many more fungi barcode from Vestadec. So they provided us new data last month, and now it's not added. Unfortunately, most of them are also not in the NSR. So that's something that is going to be maybe improved next year. Next slide. Yeah. So what we plan to do for the coming year. It's first that we have this internal dashboard that has been on the development has been on pause for like almost 2 years, but we want to actually work on the annotated the data there and improve it. We want to add more metrics and more metadata to annotate our data to have a better insight of what could be the problem in the the samples that did not work, and I'm not talking about like lab processing, but also sometimes they are like amplified. You have a file, but actually there is no consensus out of it. So we need to investigate that. And this dashboard is for now it's developed for Nanopore, but the idea is that we're going to use that as well for the metabarcoding data that coming from the eDentity program. And as well, there is a plan to do genome skimming for Horizon. So we want also to use that to visualize our genome skimming data. And the genome skimming, this is not, it has been done for BG. So we want to do that as well for Horizon. It has been, there's been like some manual work for BG because of the data are not really normalized, but we want to avoid that for Horizon. And we are going to develop a Galaxy tool. So the same way that we process the identification, the same way that we do the taxonomic identification with Galaxy, we want to do the same for the genome skimming. So first develop a tool and then use Galaxy plus our database and the APIs for that. The target list, yes. So we already work on that this last weeks. We want to add more data, more public barcodes, so that it can help us to prioritize. It's not straightforward because there is no board is follow some standards, but other databases don't. So it's a bit of work to do that, but still we're going to try to have more public barcodes. And finally, we have pipelines. They're running. It works. But there's a bit of manual work because they run on deep. They run on individual clusters. They have API, so we can see what's going on. But we have to manually connect and restart them when there's like a network problem, for example. And next year we're going to use a community developed tool that allows us to orchestrate from one single dashboard all the pipelines and restart them and look what. And also we are going to use that we now have 3 pipelines, but we will have more with genome skimming and metabarcoding.
Very much.
And that's it. Yeah.
Nice. All right. And I'll take over.
So for the few that haven't met me yet, I started in July as the new product owner of the BioCloud. And the BioCloud is for storing all of that data that we've just talked about. It's images, it's sequences, and we're talking about a lot of data. So the media items, specimens, and sequences, that was already collected. But now, as you've just heard, we've also added blasting results. So we've blasted more than 70,000 specimens, sequences. But for each of them, you get 100 results back. So we're already having 7 million records there. So we're talking about a lot of data. So that's where our expertise comes in. And what we've also added is the data of the monitoring demo sites, the talk that we started with. So you can see it's about 26 terabytes. We now have it almost fully in our own storage. The next step is actually getting it in the DSI portal that Jacob talks about, so we can actually analyze all of those images.
I need to try to connect because otherwise my battery dies.
Okay, does someone have a charger for Elaine?
Nope.
Anyone? Maybe anyone that can run outside to get one?
Yeah. If we don't screw up the audio, we should be fine.
All right, well, let's go to the next slide then. So this is the image that you've seen in July. We have sensor data, we have specimen metadata coming from ADA or from sheets, we have sequences, and that goes to the DSI portal and the DNA dashboard. Now we've added some sources. So that's the blast results. And you can kind of see it's connected because we get the sequences from the bioinformaticians. Then we add the metadata that was collected in ADA, and then we give it back to them to do the blast. And in addition, we also added sample sheets as a source. That's for the lab that goes to a batch selector tool and to the LIMS. Well, next up is that we're going to take some serious steps in order to actually publish all of that data on BOLD. In this case, it's specifically about the sequences. Well, apart from just collecting a lot of data, we're also trying to make it more robust and future-proof. And so we've added data contracts and automated data quality checks. So this is a simplified schema. You have a data source that goes into the BioCloud, and we publish it to a data consumer. And now we've actually started making contracts between the data source and the BioCloud. So that says, these are the fields that I'm expecting. These are mandatory fields. This is a text field or a number. So we define, it's really a contract that you have, but it's in code. And a nice thing is that you also have tools like Soda that then use that contract to actually perform a data quality check. So before data comes into the BioCloud, we automatically check it. We're quite far with implementing it. We've built the framework. We've added data contracts for all the suppliers in the DNA domain, and now we're adding the checks to Soda. Well, next is, for example, to make a dashboard, but also to add other domains, and also not do it only at the beginning, but also at the end, so that the people or the tools that, for example, the DNA dashboard, they also have a contract. They know exactly what they can expect from us. Yeah, and then it's not a pretty dashboard. This is a work in progress. And maybe Kevin can see in the front, in the yellow box, you can just get, did the check pass or not pass? And in the end, you can make charts that show these are the errors. It is what was correct. So you don't need to know the details, but we can make dashboards about the quality of the data, which also helps with debugging, that people that actually upload a sheet can debug it themselves. Well, and for that, we've also created a tool, because it was quite difficult, sometimes challenging, to upload sheets. So now we've created a tool where you can upload a sheet and you immediately get feedback on the correctness. Yeah. So you don't need to know the text, but you can probably imagine red on the top is an error, green on the bottom, yay. Tomorrow you will see this data in the data, yeah, in the BioCloud. And yeah, and the nice thing is that it's not just for the sheets that are used in Arise, but we've it's kind of a cooperation also with collection information. So they are working on making this available for the CRS input sheet. So the people that work at Naturalis probably have heard of that.
Thank you very much, Jessica. This one was still here. So I guess these were the updates for the teams. Hold on, because there's a little bit more. And of course, all the tools that we're creating is just so that we are finally able to have data-driven decisions on the state of our biodiversity. I see no more slides. Vincent, did you?
I didn't add any slides.
Oh, you didn't?
No.
Okay.
I can do it without slides.
Yeah, so we have Vincent Merks in the room. So, I mean, up until now, of course, you see that the product teams that were started around Arise have now transferred actually to host and deliver for multiple programs. And our latest addition is ABM. And Vincent will tell a little bit about that.
Yes. For those of you who were there this morning, maybe it's a bit repetitive, but so indeed, of course, we want to ultimately we are doing all of this, like Len mentioned, to accelerate the discovery of biodiversity, to monitor all the species. And to that extent, at Naturalis, you are maybe familiar with the Blocker schema, which is kind of this vision, okay, what should we be able to do if you really want to do that as a very high scale, focusing indeed mainly on DNA and sensors. And looking at that scheme, we had identified that it's the sensor part needs to be strengthened, right? Because Arise, I mean, we've seen today, we've developed a lot of things, but the money is ending, right? And if we really want to put it in a very high scale, and you see it generates enormous amounts of data, and so we want to do it throughout the country on a national level. We need to strengthen that. So therefore, we wrote with the whole team. Elaine was very much involved, of course, Rutger, other people in this room, the proposal of ABM, which is automated biodiversity monitoring. And so this was for the FTO program, which is the same kind of money that eDentity got funded in. So it is infrastructure money, so no scientists, only machines and people to build things. But it was successful. And so we got almost 20 million euros, of which 13 million is for Naturalis, to build this AI infrastructure to process image and sound data at a very high level. This is in collaboration with TNO, RIVM, and Rijkswaterstaat. TNO is going to focus on building kind of these stations. So ideally, you want to combine multiple sensors, and you have a station that there is also some pre-processing involved, so you don't have to send all of the data over the mobile network. And RIVM and Rijkswaterstaat, they are kind of our test cases, so they will test these stations in the field. But the majority of the money goes to Naturalis to build, to build on top of Arise, actually, these are AI capabilities. So this program hopefully will really start beginning of next year. It will take some time to secure really the funding. It's very complicated with the ministries and how this money flows, but we'll make sure that money will reach us. And then basically, I think the timing is ideal because of the Arise money is phasing out, and now we can go into this money for the whole sensor and AI part.
Yeah. Thank you so much, Vincent. And it's super cool to now see this clicking together. What we've built with Arise lives on, also through other programs, and the capabilities will stay. We are learning a lot, and we're really making progress to make sure that we can identify species quickly so that we can actually do something about our biodiversity based on use cases, based on research, but probably also very much around use cases coming from society or even industry. Thanks all for listening. I hope you had a great time. Enjoy December, end of the year. So I hope you enjoy your time there, and we'll see you next year.